All right, so today we're going to be diving into the world of insect control,
Okay.
but not the kind that you're probably picturing, like, smelly sprays or swatters, none of that.
Right.
We're talking about pgSIT, precision-guided sterile insect technique.
Wow.
That's a mouthful, I know.
Yeah.
It's a way to manage those pesky insect populations using
Yeah.
gene editing.
Okay.
And the best part is,
Mm.
it's all about precision.
Okay.
Imagine being able to target specific species
Yeah.
without harming others. Wouldn't that be amazing?
Yeah. So how does this work?
That's exactly what we're going to uncover today.
Okay.
How this PBSIT works, what the benefits are, and the challenges ahead.
Real good.
Like any new technology, there's always going to be some
Yeah.
pros and cons.
Right.
But we're going to delve into all of that.
Okay. I'm excited.
So to kick things off, maybe you could tell us what makes PGSIT different from, say, the traditional sterile insect technique.
Right. So what's really fascinating about pgSIT is that it takes the foundation of traditional sterile insect technique,
Mhm.
which we've been using for, many years,
Right.
but it adds this layer of precision that we've never really had before.
Okay.
So traditional SAT, it involved, like you said, sterilizing insects kind of en masse
Right.
and releasing them to, disrupt those breeding cycles.
Yeah.
But it had some drawbacks,
Yeah, like
like the reliance on radiation, for example, or
Mhm.
manual sorting, which could be, pretty harsh on the insects.
Right. Yeah, I wouldn't want to be the one doing the sorting.
Exactly. PGSIT is different.
Okay.
It uses this really cool two-line breeding system
Hmm.
where one line is expressing Cas9.
Okay.
And just in case, folks aren't familiar, that's that gene editing tool.
Yeah, the CRISPR stuff.
Exactly. And the other line is expressing guide RNAs.
Okay, I've heard of those, but could you explain those a little bit more for our listeners?
Sure. So guide RNAs, they basically act like a genetic GPS. They tell the Cas9 where to go, which gene to target.
So they're like the directions for the gene editing?
Precisely.
Okay, interesting.
And so when these two lines meet, when they breed,
Hmm.
their offspring inherit this genetic modification.
Okay.
And what happens is you get 100% female lethality and male sterility.
Wait, hold on. 100%.
100%.
That's incredible. So all the females die?
Exactly. It's like a built-in system,
Oh.
Eliminates the females and makes sure the males can't reproduce.
That sounds like a huge deal. Like, what are the implications of that?
It's incredibly significant for a few reasons, right? So first of all, you don't need to do that sex sorting anymore,
Oh, right.
which was super labor-intensive and not always that reliable.
Yeah, and probably pretty expensive too, I imagine.
Exactly. And then the second thing is, by only releasing sterile males, you don't need as many insects to achieve the desired effect.
Oh, that makes sense.
So you potentially reduce costs, reduce the ecological footprint of the whole program,
Right.
and minimize any, unintended consequences of releasing a ton of insects into the environment.
So it's not just about precision.
No.
It's about efficiency and minimizing impact,
Exactly.
which I think is something a lot of people can get behind.
Absolutely.
Now, I'm curious, how well does this actually work in practice?
The research is really promising so far.
Okay, give us some examples.
Let's take the mosquito Aedes aegypti, for instance.
Oh, yeah, the dengue and Zika spreader?
Exactly, notorious for those diseases.
Right.
And pgSIT has been used to actually suppress their populations in trials.
Really?
Yeah, and there's even a pgSIT strain that produces flightless females.
Whoa, flightless females.
Yeah, can you imagine?
I mean, I can, but that's pretty wild.
It is.
So that makes them even less likely to spread disease,
That's
right?
exactly. It's like an extra layer of protection.
Okay, but how do we know it's actually leading to fewer mosquitoes in the wild?
So both mathematical models and actual field data suggest that these pgSIT males, they can compete with the wild males. And that leads to fewer viable offspring,
Oh, I see.
which ultimately suppresses the population.
So it's a targeted approach, like we were talking about,
Right.
to reducing the threat of these disease vectors.
Exactly.
So much more control than just spraying everything.
Absolutely. It's about being smart about how we manage these populations.
No, it all sounds amazing, but there's got to be some challenges, right?
You're right.
No scientific breakthrough is without its hurdles.
Definitely. One of the biggest ones is getting regulatory approval.
Ah, yeah, that makes sense.
Because pgSIT involves genetic modification, obviously.
Right.
It faces a lot of scrutiny, to make sure it's safe and effective before it can be implemented widely.
So like checks and balances, safety first, always.
First.
Always.
Always.
But I imagine that can take a long time.
It can, yeah. It's a lengthy process, for sure.
What other challenges are out there?
Well, researchers are still working on, optimizing those release strategies.
Right, like how many to release and when.
Exactly. It's not a one-size-fits-all kind of thing.
Right.
Each species has its own, you know,
Quirks.
yeah, its own quirks, and you need to tailor the release strategy accordingly.
So it's like a delicate dance, figuring out all the right steps.
It really is.
Are there tools to actually track these pgSIT insects once they're released?
Yeah, that's another area of active research.
Yeah.
Scientists are developing these highly sensitive detection methods.
Hmm.
One of them is called SNSR.
What does that do?
So it tracks the presence of the engineered genes
Okay.
in the wild populations.
So it's like a built-in GPS tracker.
In a way, yeah.
That's amazing.
And it allows them to monitor how pgSIT is spreading and its impact on the target population.
Okay, that makes sense, but what about the long-term impact on the environment?
Right, that's a really important question.
Is it safe?
Yeah, and it's one that researchers are actively studying. Conducting in-depth studies to assess any potential ecological consequences, both short-term and long-term.
So it's not just a quick fix. They're really looking at the big picture.
Absolutely. The goal is to understand how pgSIT interacts with the whole ecosystem,
Right.
and make sure it doesn't disrupt that delicate balance of nature.
Like the unintended consequences we talked about.
Exactly.
So it seems like pgSIT has the potential to really revolutionize pest management.
Yeah, I think so.
But it's not just about public health, is it?
You're right. It goes beyond that.
Like what?
Well, it's showing a lot of promise in managing agricultural pests as well.
Okay.
Like, for example, the spotted-winged Drosophila.
And what does that one do?
That one's a real threat to fruit crops.
Oh, I see.
So this really highlights pgSIT's versatility, its potential to address these different challenges related to insect pests.
Multitasking. I like it.
Right.
So we have this technology that could be a game changer for both public health and agriculture. But it's not the only tool in the shed, is it?
No, it's not. You're right.
There are other approaches to insect control out there.
Exactly.
Like, some of the more traditional methods.
Right. So there's traditional SIT, of course.
Right.
And then there's the incompatible insect technique, or IIT.
I haven't heard that one.
It uses bacteria to disrupt reproduction.
Oh, interesting.
And then there are even some combined approaches using both SIT and IIT.
Okay.
But pgSIT, it offers some distinct advantages.
Okay, like what?
Think of it as a more precise and targeted approach compared to something like radiation, which is more
broad.
Yeah, more broad.
So it's like a scalpel versus a hammer.
Exactly.
Walk us through some of the key differences between these methods.
Sure. So one major difference is that you don't need to do sex sorting with pgSIT.
Right. We talked about that.
Whereas traditional SIT and IIT, you still have to separate males from females.
Which is a pain.
Yeah, it can be really challenging and time-consuming.
And remember how we were talking about that 100% female lethality?
Yeah.
That's a huge plus for pgSIT.
Absolutely.
I mean, that just makes it so much more efficient, right?
Yeah, it really streamlines the whole process.
I can only imagine.
And that efficiency translates into, potentially fewer releases, lower costs over time.
And less environmental impact, right?
Exactly. All those benefits we talked about earlier.
Okay, so we're starting to see how pgSIT really stacks up against the competition.
Yeah, and there's a lot more to explore.
I'm ready.
Let's dive in a little deeper.
Perfect.
And another cool thing about pgSIT is the flexibility in terms of release stages.
Release stages. What do you mean by that?
So you can actually release these insects at different points in their life cycle.
Right.
Like eggs, larvae, pupa, even adults.
Oh, wow. So you have options.
Exactly. It depends on what works best for the target species and the specific environment.
Okay.
Whereas other methods often you can only release adults, which can be tricky.
So pgSIT gives you more control.
Exactly. It's like a more customized approach.
Customized insect control.
Right.
Interesting.
Right.
Now, what about the cost factor?
Okay.
Is pgSIT more expensive than those traditional methods?
So the initial investment, yeah, it might be a bit higher, but in the long run, it could actually be more cost effective.
Really? How so?
Well, first of all, you're eliminating that sex sorting step.
Right. That was a big one.
Which saves a lot of time and money.
Yeah.
And then because it's so efficient, you might not need as many releases overall.
Okay.
So over time, you end up saving money.
So it's like investing in a really good tool up front.
Exactly.
That pays for itself through, like, increased efficiency.
Precisely.
Okay, I like that. But how does pgSIT affect the insects themselves? I mean, radiation, like in traditional SIT, Sometimes it can impact their fitness, you know?
Right.
Does pgSIT have a similar effect?
That's a good question. And actually, pgSIT tends to have less of an impact
Yeah.
compared to radiation. Because we're using these precise gene editing techniques,
Hmm.
we're minimizing those unintended side effects that could, mess with their ability to survive
Okay.
and mate in the wild.
So those sterile males need to be in top shape, right?
Exactly. They got to be able to compete with those wild males.
To disrupt that breeding cycle.
It's all about competition.
Now, how do we ensure that pgSIT only targets the species we want it to target? What about those off-target effects, on other insects or the environment in general?
So specificity is a huge priority here.
Okay.
And the CRISPR technology allows for really precise targeting of genes that are specific to the species we're interested in.
So it's like a laser-guided missile hitting its target.
Exactly. Minimal collateral damage.
Okay, that's reassuring.
And it minimizes that risk of, harming other organisms in the ecosystem.
That makes sense, but what if we need to change course for some reason? Is pgSIT reversible?
That's an important point, and unlike some other genetic technologies like gene drives, which are designed to, spread and persist,
Right.
pgSIT is much more controlled.
Yeah.
If we stop releasing pgSIT insects,
Yeah.
those genetic modifications will eventually disappear.
So it's like a safety switch.
Yeah, you can think of it that way.
We can go back to the original state.
Exactly, if needed.
Okay, that's good to know. I imagine there's a lot of scrutiny around using this technology, though.
Oh, absolutely.
What's the regulatory landscape like for pgSIT?
So it's still being evaluated by regulatory agencies all over the world.
Okay.
They're looking at the science, the risks, the benefits, developing guidelines for responsible use.
So it's a thorough process.
Very thorough. Yeah, it takes time, but it's necessary to ensure safety and build public trust.
Yeah, got to make sure everyone's on board.
Right.
Okay, speaking of scrutiny, let's talk about the financial side of things.
All right.
Who's funding this research?
So it's a mix of public and private investment. You have governments, research institutions, philanthropic organizations, all contributing.
Wow, so it's a collaborative effort.
Yeah, it really is.
Okay. And are there any companies involved?
Yes, there are some companies, like Agrigene Inc.
Okay, and what do they do?
They have exclusive rights to pgSIT,
Interesting.
and they're investing in its development and commercialization.
So it's not just research. They're looking at bringing it to market.
Exactly. That's the goal.
All right, now let's talk about one of pgSIT's most exciting applications, at least in my opinion.
Okay.
Its potential to combat malaria.
Yeah.
We talked about Aedes aegypti, but pgSIT is also being studied for Anopheles gambiae, right?
That's right. That's the main malaria vector.
I mean, if this could actually work against malaria, it would be huge.
It would be absolutely transformative.
Right.
And for AZIT to be successful against any mosquito species, there are certain criteria that need to be met.
Okay.
The sterile males have to be competitive.
Right.
The technique needs to be scalable for mass production, and it needs to effectively reduce the target population.
Okay, and does pgSIT meet those criteria for Anopheles gambiae?
It does. It actually checks all those boxes.
Really?
Yeah, which is why it's so promising.
So it's like pgSIT is tailor-made for tackling malaria.
It really seems that way.
Have there been any field trials?
Yes, there are ongoing research efforts in several countries,
Yeah.
and the early results are very encouraging.
Give us some examples.
So studies have shown that pgSIT can significantly reduce Anopheles gambia populations
Wow.
in controlled settings. And mathematical models predict that larger scale releases could have a major impact on malaria transmission.
I mean, that's incredible, but I imagine there are still challenges specific to using pgSIT for malaria, right?
You're right, every mosquito species is different.
Yeah.
One of the key challenges is optimizing those release strategies again,
Okay.
because their behavior, their habitat preferences, their mating patterns might be different from, say, Aedes aegypti.
So researchers need to figure out the right approach for Anopheles gambia.
Exactly. Adapt the strategy to the target and the environment.
It's like a constant process of refinement.
It is.
But beyond those species-specific challenges,
Yeah.
are there any broader ethical or societal considerations around pgSIT?
Of course.
We're talking about, like, genetically modifying entire populations. That's got to raise some eyebrows.
Absolutely. Responsible innovation is paramount here.
Right.
And there are ongoing discussions about the potential ecological and ethical implications.
Okay, so it's not just about the science.
It's about the societal impact as well.
Like, what are some of the questions that are being raised in those discussions?
So one of the main concerns is the potential for unintended consequences, right?
On other species or the environment?
Exactly. We need to make absolutely sure that pgSIT is specific to the target and doesn't disrupt the balance of nature.
So it's like walking a tightrope, advancing the science, but also being mindful of the risks.
It's a delicate balance.
And how are researchers addressing those concerns?
Through rigorous testing and monitoring.
Okay.
Extensive studies to assess the impact on non-target organisms, both in the lab and in the field.
So they're not taking any chances.
No, they're being very thorough.
And they're also exploring ways to make the technology even more precise.
Right, and more controllable.
To minimize those risks.
Exactly. It's an ongoing process of improvement.
Now, what about public perception?
Okay.
How are people reacting to this idea of using genetic technology to control insects?
Public acceptance is crucial for any new technology.
Right.
And there are often, concerns and misconceptions around genetic modification.
Yeah, people hear GMO and they freak out.
Exactly. So transparency and public engagement are super important here.
So it's about educating people.
Yeah.
Addressing their concerns.
And involving them in the decision-making process.
Okay, so it's a collaborative approach.
Absolutely. We need everyone at the table.
Now, let's shift gears for a moment. We've talked about disease vectors, agricultural pests.
Yeah.
What other applications are there for pgSIT?
Well, while those are the main focus right now,
Right.
researchers are exploring other areas as well.
Like what?
One area that's getting some attention is invasive species control.
Oh, interesting. Tell me more about that.
So imagine using pgSIT to target invasive insects
Okay.
that are, disrupting ecosystems, harming biodiversity. It could be a game changer for protecting native species and preserving those delicate ecological balances.
Yeah, invasive species are a huge problem.
They really are.
So pgSIT could be a powerful tool in that fight.
Exactly.
So we're talking about protecting human health,
Yeah.
safeguarding our food supply,
Right.
and now even preserving biodiversity.
It's pretty amazing.
It's like pgSIT is a Swiss Army knife of insect control.
It really is. It's incredibly versatile.
Okay, but the most exciting part to me is that we're still in the early stages of pgSIT development.
Absolutely. We're just scratching the surface.
As research continues, we can expect to see even more creative and impactful applications.
Definitely. The possibilities are endless.
It's like we're at the beginning of a new era
Yeah.
in pest management
One that's driven by precision and innovation.
and a deep understanding of genetics.
Exactly. We're harnessing the power of nature itself.
But with this power comes responsibility.
Yeah.
We need to use this technology wisely and ethically.
Absolutely. That's the key.
So it's not just about the science. It's about our values.
It's about our responsibility to the planet.
Okay, so final question for you.
All right.
We've learned about pgSIT, its potential, the challenges.
Yeah.
Where do you see this technology heading in the next decade?
It's always tough to predict the future.
Right.
But I believe we'll see pgSIT move from research to real-world implementation.
Okay.
I think it'll be used to combat a wider range of pests. Those that transmit diseases, damage crops, disrupt ecosystems.
So from the lab to the real world.
Exactly. Tackling real-world problems.
Well, that's exciting.
And I think pgSIT will play a key role in creating a healthier and more sustainable future.
A future where we can live in harmony with nature.
Exactly. That's the goal.
But there will be bumps along the road, I'm sure.
Oh, definitely.
What are some of the hurdles that need to be overcome?
So regulatory approval is a big one.
Okay.
Navigating that landscape can be tricky, but as the science advances and the safety is proven, those pathways will become clearer.
So it's a matter of time and evidence.
Yeah.
What else?
Public acceptance is another key challenge.
Right, we talked about that.
We need to overcome those fears and misconceptions around genetic technologies.
Through education and transparency.
Exactly.
And showing that pgSIT is safe and effective when used responsibly.
It's all about responsible innovation.
Okay. Any other potential roadblocks?
Well, cost is a factor.
Right.
Developing and deploying pgSIT at scale, it can be expensive.
Okay.
So we need to find ways to reduce those costs.
To make it accessible to everyone.
Especially to those who need it most.
So it's a balancing act.
It is.
Innovation, regulation, public perception, cost effectiveness.
It's a complex equation.
But one that's worth solving.
Absolutely.
Given the potential benefits of pgSIT.
I agree. And I believe that with continued research, collaboration, commitment to responsible innovation.
pgSIT has the power to revolutionize pest management.
And create a healthier and more sustainable future for all.
It's an inspiring vision,
It is.
and one that I hope we can achieve. It's really fascinating to think about how this could actually change things.
Yeah.
Our relationship with insects, moving away from battling them to
Right.
managing them.
More harmoniously.
Exactly.
Yeah.
But let's come back to the present for a second.
Okay.
Is pgSIT actually being used anywhere right now?
So it's still mostly in research and development, but
Right.
there are some real-world trials happening.
Really?
Yeah. For example, there's a company called Oxitec.
I've heard of them.
They've been conducting trials with a genetically modified Aedes aegypti mosquito
Okay.
that carries this self-limiting gene.
So it's not exactly pgSIT,
Not strictly.
but similar principles.
Right. It's still genetic control.
And what have the results been like?
Pretty positive.
Okay.
They've seen reductions in mosquito populations
Hmm.
and the spread of diseases like dengue.
So it's actually working.
It's looking promising.
That's exciting. So what will determine how quickly pgSIT becomes more widely used?
Well, cost is a big one.
Yeah, makes sense.
It could be cost-effective over time,
Right.
but that initial investment can be significant.
Especially for large-scale programs.
Exactly.
And I imagine that cost can be a real barrier for, regions that are hardest hit by insect-borne diseases,
Absolutely.
but might not have a lot of resources.
Right. And there are initiatives to address that.
Okay, tell me more about that.
So there are international organizations, research institutions, philanthropic groups working to make pgSIT a reality in low-income countries.
That's great to hear.
They're looking at new funding models, partnerships, building capacity.
So it's a global effort.
It is, because this technology should benefit everyone.
I agree. Now, what about the research?
Yeah.
What's on the horizon for pgSIT?
A lot of exciting stuff.
Okay, give me some examples.
Well, they're working on developing even better genetic constructs.
So making pgSIT even more efficient.
Yeah. More precise, adaptable to different species.
Okay.
Imagine being able to tailor it to specific environments.
Wow.
Or even target multiple pests at once.
That would be incredible.
It would really change the game.
And what else?
They're also working on more sophisticated monitoring tools.
Oh, right. Like those genetic tracking systems.
Exactly, making them even more sensitive, accurate, cost-effective.
So we can get a better picture of what's happening.
Kind of real time.
And fine-tune the strategies.
Exactly.
So it's all about constant improvement.
It is. We're always learning and refining.
Well, we've covered a lot of ground today.
We have.
The science, the applications, the challenges, the ethical considerations.
It's been a fascinating deep dive.
It really has, and it seems like pgSIT has incredible potential.
It does.
But it also raises those important ethical and societal questions.
Of course, any powerful technology does.
We need to be mindful of how it's used.
And who makes those decisions?
Right. It's about establishing ethical guidelines
And frameworks.
that involve everyone, scientists, policymakers, communities.
Everyone needs a voice.
So the use of pgSIT aligns with our values.
As a society.
Are there any efforts to address these challenges?
Yes, definitely. There are international collaborations and working groups
Okay.
developing ethical guidelines and best practices.
For pgSIT and other gene drive technologies.
Right, ensuring responsible development and deployment.
Transparency and consent are key.
Absolutely.
Well, it's been a truly thought-provoking discussion.
I agree.
And pgSIT is clearly a game changer.
I think it has the potential to transform pest management.
But, as we've discussed, it's crucial to use it wisely and ethically.
With respect for the balance of nature.
And on that note, I think we've reached the end of our deep dive into pgSIT.
I agree.
It's been a fascinating journey.
It has.
And I hope our listeners found it informative and engaging.
Me too.
But remember, this is just the start of the conversation.
Right.
The field is constantly evolving.
New discoveries all the time.
So stay curious, keep asking questions, and engage in the dialogue
It's an important conversation to have.
about shaping the future of this technology responsibly.
We all have a role to play.
Perhaps one day we'll look back and see this as a pivotal moment
Yeah.
when the potential of pgSIT first came into focus.
That would be amazing.
Until then, keep exploring, keep learning.
And stay tuned for the next deep dive.
We'll see you then.